Welcome to this week's WIP, everybody. For this week's work in progress, we have Dr. Sam Telford. He is an epidemiologist focusing on vector-borne infections, mainly those transmitted by ticks. His 40-year career has contributed to the development and approval of the first human Lyme disease vaccine, incriminated new agents of tick-borne disease, and described the ecology and epidemiology of these infections. Dr. Telford's laboratory is currently funded by NIH grants, and he advises local, state, and national organizations on public health interventions against tick and mosquito-borne infection. Welcome, Dr. Telford.
Well, I'm really delighted to be here and really sorry not to be an official member of NUVEC, but I fully support what you're doing. Today, you will hear something different. It's not intended as a pitch for a magic bullet. It is no such thing, but nonetheless, it is something new and maybe the wave of the future. And so I don't need to tell everybody online about the importance of coming up with interventions for Lyme disease and the other deer tick and increasingly the Lone Star tick transmitted conditions, infections, including conditions such as alpha-gal. They continue to expand their range. They locally intensify because of suburbanization. Our demography is changing. It means that, a lot of people wave their hands about climate change. It's not climate change. It's like the incidence of these things depends upon who appears in the emergency rooms and gets reported. And our people over 60, and that includes me, and I believe it includes Steve there, we're much more susceptible than we used to be to having an adverse outcome due to things that we once would have just sort of laughed at, or relatively speaking anyway. So our demography is driving changes in the incidence as is suburbanization. Deer abundance continues to increase, and I remain the nation's biggest advocate for killing deer. To try to intervene both against the deer tick and the Lone Star tick. But nonetheless, the fact is incidence is increasing and it's locally intensifying as well as expanding its range. And therefore, we really need to just do something about it. And so you've given me this soapbox, and every time I have a soapbox, I try to point some general concepts out. Public health interventions have short-term and long-term modes, and a lot of people don't really understand the distinction. They're not mutually exclusive, but short-term modes, you need to have these done every day, week, month, forever. It's like a monkey on your back. And in order to get people to continue to do this, the results have to be quick and clear. And the scale can be multiple. It can go from personal, individual protection to trying to do something for the community. And these include things like repellent, spraying, burning, vaccination that requires boosting, like all the Lyme disease vaccines that have been developed. But long-term modes are what we really need to try to focus our attention on, where we invest a lot of time and money and energy right up front. But then the maintenance is relatively little for the future. But the big difference is that our goal is to reduce risk over time. You can think of it sort of like tobacco cessation, that it took forever. It took like 35 years to get smoking rates down to what they are today. But it's been a fantastic public health achievement. The scale can be large. Communities, areas, regions, and I maintain that deer reduction is a long-term mode of intervention. And these are all, laid out in the tick management handbook. But if I had an advice for Kirby and making a new edition, it'd be like, make sure and emphasize this short-term versus long-term approach to intervention. And so we all know about the modes of intervention that we're now recommending, even though CDC has said none of them work. Repellents, intoxicants, and clothing, and showers, and it's like there are all of these things that can be done, but setting a timer here because I tend to yak a lot. Habitat avoidance, and the centerpiece, I think, is the insect, the the permethrin treated clothing, and in particular the socks. And I like to show this slide because a picture is worth 1,000 words. I'm trying to disseminate this. The CSTE, I believe, is going to try to push this image out fairly soon. But on the left is a Lone Star tick larval infestation, several patches of larvae that I hit out on Tuckernuck Island, and the right side are my feet. And I did the experiment, one sock on permethrin sock on one side and no treatment on the other, and you can readily see what happens if you do this. And so I think this kind of image speaks volumes to the public and reinforces our recommendations. So but that's a short-term intervention. If you don't wear the socks the next day, you will get bitten up just like I did on one of my ankles. But what can we do for long-term interventions? And I think yeah, there are little variations in the story, but in general, the main drivers for the emergence of Lyme disease as a problem in the eastern U.S. is deforestation and reforestation, increased development, and burgeoning deer herds. And of those three, we can only attack two of them. Increased development and recreational use, we can never do anything about. Deforestation is something we haven't really talked about, and I'm starting to think, maybe we ought to recommend that we chop down a bunch of trees, pretty much in line with what Mr. Trump wants to do. And I still pound on the table about burgeoning deer herds, but are there other possibilities for long-term intervention? All of these integrated they have to integrate with short-term methods. Habitat management is a short-term method. Education and awareness, unless you keep it up, short-term method. Spraying is not a dirty word. We should be using it more and more. And the host-targeted acaricides, again, all short-term interventions. Deer reduction is not a short-term intervention. If you expect to kill deer and have a risk reduction the next year, you don't understand why you're killing deer. So, what are long-term solutions? You can attack on two fronts, preferably on both fronts. But they're largely independent factors that can be attacked, and it's tick production and infection of the tick. You can attack the reproduction of the tick by trying to prevent eggs from being laid. The source of the reproductive blood meal is always a large animal. Thank God it's not a small animal. And so deer reduction targets tick reproduction because adult ticks do not feed on the small animals. And it's remarkable how misunderstood this concept is. Every time I speak about deer reduction, everybody says, oh, but it's really the mouse that's the culprit. Well, yes, it is for point number 2, but it's not for point number 1. And it's something we need to do better in terms of communicating to the public as to our possibilities for intervention. We can attack the infection of the tick. That uninfected larvae need to get infected. They don't inherit an infection. And I'm speaking about Lyme disease pretty much, but Babesia, microti, and Anaplasma, Figs tofflum are not inherited by the tick. Borrelia miyamotoi certainly is, and Powassan virus can be, but it's not clear how risky a larval bite might be or whether it actually contributes to perpetuation. But infection can come from a number of different animals. That's, everybody says that is the case. Larvae do feed on other animals, large animals like deer or cats, but they certainly don't get infected from them. And so this idea of genetically modifying mice, it's targeting tick infection. It is not targeting reproduction of the tick. And so a little background about this idea of genetic modification, and it still has its basis. It's really gratifying to me that something I worked on as a young scientist is coming back and still being valuable to potential interventions. Errol Fickrig and I had little friendly argument about the adequacy of a syringe challenge for testing the effects of a vaccine. When he and Rich Flavell came out with his paper in Science, Errol had never done a science experiment in his life. He was a medical student. He got his MD, got a fellowship for a internship at Yale New Haven Hospital, and said, "Oh, I want to do research." And he knocks on Rich Flavell's door. Flavell is one of the top immunologists in the world. And he looks at him and he goes, "Well, we have this little infection called Lyme disease in our backyards. Why don't you make a vaccine for it?" This is 1989. Alan Barber had just published the sequence for the outer surface protein A. For those of you who don't know, this is a spirochete, the Lyme disease spirochete in cross section, and it's covered with a membrane of various outer surface proteins, lipoproteins, and the most immunodominant one, the most highly expressed one in culture is outer surface protein A. Alan had just published the sequence for OspA, and Errol devises PCR primers, amplifies OspA from strain N40, buys a Pharmacia GST fusion kit, follows directions, expresses OspA, immunizes mice, and then challenges them with syringe challenge with cultivated N40 spirochetes, and he gets a science paper out of it. And all of his fellow graduate students, all of his fellow postdocs just hate his guts because it's his first experiment and he gets a paper in Science. And we at Harvard, we were like scratching our heads going, well, but if you used a tick, we've got these immunosuppressive and anti-inflammatory compounds and tick saliva, it'll never work. And to his credit, Errol said, "Come on down." I brought ticks. I personally placed them on mice that were coded. Only he knew the code. And he sent the engorged ones back to me. I dissected them, and I'm going, what the hell happened? Because half of them had what were very clearly intact spirochetes by immunofluorescence, and the other half, 50%, had all these clusters of antigen. I'm going, how did I screw up? Because I brought down ticks that were 100% infected, and I'm only seeing 50% infection. And then the aha came to me, which was we've been working on reactivation and the possibility that there was OspA that was being targeted by the vaccine coming into the blood meal and killing spirochetes before they had a chance to get out. And subsequently, 2 years later, Tom Schwann and Joe Peisman published the paper on the differential expression of OspA and OspC and during the course of feeding, and it all clicked. It all made sense. And with that, it was clear that anti-OspA immunity acts before reactivation of the spirochete and prevents the antigenic shift to OspC, and therefore OspC being critical for transmission, aborts transmission. I was interested in this because we were puzzled as to why anti-OspA antibody was rare in wild populations of animals, but interested in the potential for using antibody against OspA as an intervention. And this experiment was done with passive transfer, where you could immunize mice against OspA, take their serum, and then put that antibody into mice and see what happens to the spirochete. So if you take infected mice and do xenodiagnosis on them, you get 83% of the mice yielding infection. But once you infuse them with antibody, that infection rate goes down to 25%. And when the antibody clears, if you use heterologous antibody, so you use white mouse, laboratory mouse antibody and put it in a paramescus, it disappears over the course of 2 weeks or so. The infectivity is restored when the antibody goes away. And so it was very clear that antibody was critical in the initial events of colonization of the colonization of the spirochete and the tick, or during the course of the blood meal when there's a shift to OspC. And Errol and I actually put in a patent application for oral immunization of mice with OspA. And Harvard in its wisdom and Yale in its wisdom said, we'll never get our money back on this, forget it. But nonetheless, this very powerful mode of action convinced SmithKline to go ahead with a vaccine that had been developed by Ulrich Scheibel and Markus Seemann in Germany, which was essentially, it had the same mode of action. It was anti-OspA. It was a recombinant OspA, and I was privileged as a postdoc to run the phase 2 trial for SmithKline on the islands off the coast of Massachusetts. And then Alan Steer ran the phase 3, and it's important to note that Lymerix is the only intervention proven to reduce the incidence of Lyme disease using very stringent criteria. The CDC likes the randomized placebo-controlled trials, and that's why most of the things we recommend have not been shown to be effective, is because there have been no randomized placebo-controlled trials of a large enough scale to have statistical significance. But so this is the only intervention that we know of that reduces the incidence of Lyme disease. But if you look at it, the vaccine efficacy over 2 years is not spectacular. It's, 50% or so over 2 years. And you do need yearly boosts. It requires the prime and 2 boosts for the first year, and then a boost before tick season of the second year, and subsequently as well. And so a lot of people look at that vaccine and say, well, it wasn't very effective. Well, another pontification slide, public health versus medicine. Public health focuses on populations. Medicine focuses on individuals. Public health has an emphasis on reducing morbidity and mortality for groups, whereas medicine focuses on the individual. And public health tries to do the most good for the greatest. But the physician must do everything possible for the individual. The time scale for public health is of longer duration, whereas for the individual, it has to be immediate. And so when you look at things like vaccine efficacy or treatment, to a physician, a 50% effective treatment is not really worth the effort. But for a public health person to reduce incidence by 50% over 10 years, that's a major public health achievement. And that's where the promise of vaccination or other strategies over time might be valuable. And so that's what I mean by long-term intervention. So this talk is focused on mice, and shame on us. At Harvard, we pounded on the table about Lyme disease spirochetes being mainly maintained by white-footed mice, based upon largely the paper by Jay Levine, but subsequently Andy Spielman, using his bully pulpit at Harvard, pounding on the table, saying that it is white-footed mice. If you look at all of the trapping that we've done on the islands, although this was at Ipswich, not an island, 85% or more of the larvae that we collect are of the animals that we collect are white-footed mice, and 95% of all the subadult ticks, deer ticks on mice are on mice, as opposed to other animals that were trapped. And their reservoir competence is very high as well. And if you ask the AI, of course, it says white-footed mice are the primary host of the bacteria that cause Lyme disease. And the Curie Institute, Rick Osfeld and his people have been the main movers and main proponents of this idea that white-footed mice are the main target for the main source of infection for ticks, regardless of the fact that they do indeed, in their modeling, demonstrate that white-footed mice are not the primary host for either the tick or for the Lyme disease bacterium. But nonetheless, there is this pervasive attitude out there that white-footed mice are the main hosts, and if you attack them, something will happen. This is seen with things like Damonix tubes or tick tubes. It's seen by oral OspA vaccines marketed by US Biologics. And so there is this focus on the white-footed mouse. Well, when Kevin Esvelt, a bright young bioengineering faculty member at MIT, approached me in 2017 and said, I want to CRISPR some mice and make them resistant to Lyme disease and release the mice and reduce the risk for Lyme disease. He actually used the word eradicate, which it's a forbidden word with me. I said, well, we've been working now, many of us, for 30 years, 40 years, trying to reduce the risk of Lyme disease with all of these interventions that I just talked about. And we've gotten nowhere. It's just disgraceful that the incidence and intensity of infection has increased over time as opposed to being reduced. And so I said, this is a completely new idea, and I'm happy to try to help you. Joanna Bookthall was his graduate student. Thank God, after 7 years, she finally got her degree in February after me pounding on the table all the time about getting it done. She's done the bulk of the work with this experiment, but this idea is solely Kevin's. I am here. I am part of the group to try to bring some perspective to it and to see if there are ways of making it happen. Not only that, but the approach that we're taking depends very much upon what I did as a young scientist, that is anti-OSPE immunity. But all the science aside, Kevin came out of George Church's lab and it is one of the world leaders in the responsible use of these powerful technologies, realizing that things like genetic modification of animals, once you let the genie out of the bag, you're never getting it back, and therefore it's a powerful step in changing the environment. And therefore, just as with a clinical trial, there should be some degree of informed consent among those who might be affected by this change in the environment. And so this was another really appealing aspect to me. I mean, we're seeing some of this now with the release of genetically modified mosquitoes in South America and West Africa. Where there are information sessions, are local people on advisory committees, and there is a process of informed consent. But at the time, 7 years ago, 8 years ago, it was still not something people think. They put the science first, and then, well, what do we do once we get the tool and want to develop it? Can we just go in and put them out there? I mean, there is a reason why the first trials of genetically modified mosquitoes were done in Mexico and in West Africa, as opposed to the US. They'd be held up in litigation, is my guess. But nonetheless, Kevin was very adamant about having community input or at least transparency from the very start. He had a website on responsive science. We developed a steering committee appointed not by us, but by the Board of Health on Nantucket. They are kept apprised of the developments of the project. And eventually, if it goes to a field trial, we'll help develop the study designs. And, there will be go and no-go decisions that they will be capable of, just as with a data safety monitoring board would have that power during a clinical trial. So there would be evaluation points after the mice are made, and we have some semblance of a good workable plan for introducing mice. And of course, the appropriate regulatory approvals, which is not, probably going to be just as difficult as making the mice themselves. Then the Data Safety and Monitoring Board will look at the accumulated data and make recommendations to the Board of Health, and try to find a small release site, isolated release site where we could do a limited field trial to accumulate all the data that's going to be needed in terms of environmental impact for regulatory approval. And then a larger release would have to be approved by the whole community. In the case of a place like the Vineyard or Nantucket, this would be at a town meeting. And so there is this process, and it's interesting to note that his appointment is primarily in the media lab, where they're interested in changing the way people think about things. So they're not hard science, but they use hard science to try to make have people make decisions. And so this was fully, this is what he spent a lot of time on as a result of his appointment at the Media Lab, but he also has an appointment with bioengineering, and that's where this comes in. So I argue, and he argues now, that this is an alternative to short-term methods such as baited vaccines or drugs or killing mice or using tick tubes. Heritable immunization, and interestingly, this heritable immunization concept has application on a broader scale. I mean, imagine, yes, there are ethical issues right now. 10 years from now, I argue that those ethical issues will have vanished into thin air. But can you imagine kids being born expressing antibody to the major childhood illnesses instead of being shot up and down in their first several years of life with vaccines that induce those very antibodies? And so I see this as the major contribution to knowledge that Kevin is doing with this project, that we're doing with this project, is heritable immunization, but using a system where it might have public health utility that is less ethically challenged than looking at humans. Obviously, there are ways of trying to use this for example, with poultry flocks and avian influenza, with cows and brucellosis and things like that. But heritable immunization, the proof of concept will be with Lyme disease. So he proposed making so-called tick-borne disease-resistant mice. I argue that it's rendering them non-competent, there's no disease there. But to target a main reservoir for Borrelia burgdorferi, and obviously we're focusing on mice because mice are convenient to work with, and everybody says mice are the main source of Borrelia burgdorferi, at least in the eastern US. So the idea is to vaccinate mice. And when I vaccinate white-footed mice in the lab, make antibodies, have them make antibodies, pull their spleens and bone marrow, and clone out the antibody encoding genes and make recombinant antibodies. Test those antibodies by neutralization. You notice there's tick saliva in there. We do realize that what we're making is a platform. We're using Borrelia, and in particular OSPA, as the target. But if there's ever an antigen or antigens that are capable of inducing anti-tick responses, that platform, like some of us are old enough to have had 8-track and cassette machines. The cassette can simply be replaced with an anti-tick antigen into the system. But to use the most neutralizing of these antibodies, recombinant antibodies, and then CRISPR the mice, make it so that they're heritably immunized and producing from birth antibody against Borrelia burgdorferi, and then find ways of introducing them for a small field trial, and then eventually spread it to larger areas. So the objective is to prevent infection of the ticks. If you can't protect the mice as if you were vaccinating the mice, you can render them less infectious by the fact that anti-OSPA antibody is coming into the feeding larva. Even if the mouse is infected, that tick is less likely to be infected when it molts to a nymph. We use LA2, the monoclonal antibody LA2, as our proof of concept. That's the thing that we're using to modify mice at the moment. It is well known to neutralize Borrelia burgdorferi sensu. It was developed by Ulrich Scheibel and Marcus Seemann as part of their vaccine program in the early 90s in Germany. And it is so good, it was used as the FDA neutralization standard for the SmithKline-Lyme clinical trials, people were considered to be adequately protected after vaccination if they had sufficient LA2 equivalent in their serum. So this is an early slide, the timeline of phase 1, where you're engineering mice, you're preparing for the field trial. So much of this has been done. We have done this. I vaccinated mice, and Wayne Marasco at Harvard Medical School, very, very prominent immunologist, we've got probably 24 paramiscus recombinant antibodies that neutralize very nicely in vitro. So we're ready to go, but right now we're using an LA2 to engineer mice. And, obviously the first thing to do is proof of concept. So using laboratory mice have been modified to express LA2. And they are, as I'll show, fully protected. And now the bigger challenge is to actually modify paramiscus, which are not mice, but rather they're chrysetids. They're much more like hamsters than they are mice. And so all of the transgenesis methods that everybody routinely use are not necessarily applicable to a paramiscus. In fact, we couldn't even determine the moment of ovulation for paramiscus, even though that's routine in mus. And that set us back several months trying to figure out when paramiscus ovulate. And once that was achieved, then things could proceed. So we put out a preprint on this. It's the result of 5 years of work on heritable immunization, we had to go to single chain variable SEFV antibody instead of full length antibody, simply because of the expression site not accepting that large a piece of DNA. But there's a safe harbor locus ROSA 26 that is well known for transgenesis and mus, and so that was routine. And the DNA was delivered by pronuclear injection and transplanted to pseudopregnant females, and litters were made. And once you have the first litter, then it's selective breeding. And we now, or MIT now, I have them here too, has several generations worth of mice, either heterozygous or homozygous, for expression of OspA-SCFV. The heterozygotes will express a certain amount of LA2 equivalents. Obviously, the homozygotes do very, very nicely, and the wild type mice express nothing. We have tested these by looking for the effect that Errol and I saw very clearly with his vaccinated mice, by putting infected ticks on these mice that were born, this probably third or fourth generation, filial generation after transformation, and testing the sera for antibody, as well as looking for this very clear phenotype of destroyed spirochetes in the gut of ticks that had engorged on mice. And interestingly enough, there's pretty good antibody levels in mice I don't understand this. Anyway, the number of ticks coming off of mice that were infected were no different between the challenge groups, and then you had antibodies in mice that were expressing anti-Borrelia antibody in their serum. And these were all normalized to control, so it's really the inverse. The ones with low levels are actually showing protective levels, and the ones with nothing, as expected for wild type, are shown at higher OD levels here. So it's actually a competitive ELISA that's being used to determine antibody. And so we were using xenodiagnosis to express, to determine the efficacy of this expression. And so the mice that were challenged with infected ticks, we waited 30 days. A lot of people only do their xenodiagnosis a couple of weeks after infection. We wanted to give infection the best chance of being established and waited 30 days, put larval ticks on, and then determine whether nymphs molting from those larvae became infected. And there was complete protection of modified mice, including heterozygotes, from infection, and thus their infectivity was reduced. And so these mice failed to infect the next generation of ticks. Even those that were infected, there were breakthrough infections in the heterozygotes. They were infective to the ticks, but the Borrelia did not persist until the nymphal stage. And so we argue that the transmission blocking produced by this modification is expressed in two ways. One, you can outright protect the mouse from infection, but you can also, if there is a breakthrough infection, those ticks will not retain, the larvae that feed on them will not retain infection to become an infectious nymph. And so proof of concept has been established. Now, what's the fly in the ointment? And as I alluded to very early on, well, everybody says white-footed mice are the end all and be all. And over the years, I've been puzzled to see all of this. There's never any consistent evaluations. Damonix tubes, they work in some people's hands and they don't work in other people's hands. And it doesn't matter what intervention you're doing against mice. It's never fully effective as expected. And it just goes back to the possibility that there are many other hosts out there contributing infected ticks. And so mosquito biology was greatly enhanced by the ability to do host blood meal analysis. And you're able, especially the human malarial epidemiology, the breakthrough was to use a host blood meal analysis and determine whether mosquitoes were anthropophilic or not. And so it's been a goal that has largely eluded everyone. And Heidi Gertek came up with the great idea from forensics of using retrotransposons, which are present in many uniquely in many groups of animals. So you're not going to get retrotransposons that are specific for like P. leucopus versus P. maniculatus, but for Peromyscus and other things related to Peromyscus. So developed a retrotransposon-based PCR assay for host blood meal remnants. Deer ticks. Ticks are very different from mosquitoes in that there's complete intracellular digestion. There's very little material left after the tick molts, and it disappears quickly after the molting process. And so the very early results she was getting, we were seeing some effects like this where we could not identify, these were ticks from Tom Mather, where we could not identify the blood meal, yet we were using primers for all the major animals that we thought likely to serve as hosts for deer ticks in these sites. And it turns out Tom had left the nymphs that he'd collected for us on his desk for a couple months, and they aged, and there was no DNA left to identify. And so once we knew that as an issue, and also a very astute reviewer of one of our papers, who may actually be on the call, made the point that, well, there's environmental DNA too, and maybe you need to take that into account when you're doing these host blood meal analyses, given the fact that this assay is so sensitive. And so when we started bleaching the outside of the ticks to remove environmental DNA, things were clarified even more. And so now I think we've got a pretty good assay, with the caveat that if you get ticks in July or August, they're really not going to give you much signal. You got to get them fresh when they first appear in end of May and through June, and freeze them right away, and then bleach them, and then do the analysis, and then you'll start getting good data. And it turns out that even in places where we would expect the mouse to be the main source of infection, like Nantucket, which has a depauperate fauna, it's things like shrews, which are infecting just as many ticks as mice. And if you look at Martha's Vineyard, it's the same way. And I think it really depends on the site and the time. It could change from year to year. We've shown that on Nantucket. So between sites and between years, the contributions of mice will be different. So does this really mean that we're just not, what we're doing is all academic? That we shouldn't really look at mouse targeted intervention as a real public health possibility. But no, I don't think so. I think there is certainly places where it is mouse driven, and Rick Osfeld insists that Westchester County is mouse driven. So that's great. Well, maybe we can do a trial there. But it really should be considered another. Andy Spielman used to use the term quiver of arrows. We need a lot of arrows in our quiver so we can apply as many of them towards our target as we can. So it's part of integrated tick management. It's a tool that a community can choose to use or not. It may contribute to reducing risk. I think we need some modeling, good modeling, to look at what reducing mouse contribution to force of transmission may have on cumulative effects in the future. There's been some modeling, short-term modeling done. For example, Jean Sao did this for her Lyme disease vaccination of work, OSPE vaccination work, as her thesis project, and showed very nicely that even with a vaccination rate of about 50%, you could actually impact over time the enzootic transmission cycle. So where are we going with this? Where are we now? We have mus musculus that are heritably immunized. There have been some good steps in transgenesis of Peromyscus. There's some GFP Peromyscus that sadly didn't make it to adulthood. They were cannibalized, so there was a little issue with the MIT mouse colony having to go down for a while because of dirtiness. But I think that's all back on track. We need, it's expensive work, so we need a little more money for that. But nonetheless, we expect within the next year or so to be able to validate and test Peromyscus transgenesis. And once we get those mice, it'll be easy to provide proof of concept that they're protected, just like mus are. We will use the recombinant antibodies that Wayne Marasco made, Peromyscus antibodies, not LA2, which is mus. And then big question is, can you select for resistance mutations? And that's why we needed a suite of recombinant antibodies, is that they target different epitopes, and by incorporating antibody with 4 different antibodies being expressed at the same time, maybe we can reduce any chance of there being resistance cropping up. Obviously, we need to find ways, what kinds of potential environmental impacts will there be? I don't think there will be any. It's sort of like eating genetically modified salmon. Well, if a weasel eats a genetically modified mouse, so what? I think it's going to be tough mapping out the regulatory approval strategy. Which agency will take charge of this, and it's very likely we'll have to consult everybody, the EPA, the USDA, the FDA, CDC, and only then will local authorities, our Department of Public Health and our Massachusetts Division of Fish and Wildlife, once the federal authorities have examined this, then maybe they would consider a permit. And most importantly, I think we need to test methods for introducing mice. Kevin and I have gone back and forth on this. There's no way in hell we're going to do inundative release of Peromyscus on islands, and not multiply anyway. We're going to have to do things like be clever about it and hit them when the population is low. In these sites, we have a boom and bust type demography where the summer's population of mice are really the result of breeding by a few founders in the spring. And so if we attack and if we release in the spring, I think that we can get away with relatively few mice. I'm actually of the opinion that laboratory reared mice are less fit and will never make it. So we have to find ways of doing all of the to actually target males and find some way to alter their sperm so that we can do this, give them a shot in when we trap them and release them and do it that way. And we need cryopreservation for Paramescus. Apparently, Jackson Labs has tried and they failed. And so we need to find ways of protecting our stock because it costs money to keep these guys around. But I'm a researcher. I want to see basic questions and I'm puzzled with the protection that we see with heterozygotes, which are not expressing a lot of anti-OSPE antibody. And Mark Klippner saw this with the humanized monoclonal OSPE monoclonals that he's been looking at for clinical trials. That sometimes the neutralization titer gives them misleading results, and you can get efficacy with low levels. And they hypothesize that there is some blocking by anti-OSPE antibody in terms of the switch to OSPC. And so we're looking at that right now. I'm OSPE only has 6 serotypes across the world, so it's clearly not under selection or intense selection. But if we put modified mice out there, are we going to place OSPE under selection, and will we see the generation of new serotypes? I think that's a really interesting question. And then most important of all, I grew up as a tropical public health person. I still retain an interest in tropical public health. This platform could be used to attack rodent-maintained zoonoses such as lepto or hantavirus or the arenaviruses. I mean, you can imagine being able to do this in West Africa with Lassa fever, where other kinds of interventions might not work as well. And so that's really where my interest lies. And this is like work, 10 years worth of work that I've outlined here that will keep me going well into past my expected years of retirement if I'm not forcibly retired by lack of funding due to current funding issues. And so with that, this work's been founded by an R01 from NIH, a multiple PI R01 with famous names like Wayne Marasco and Rudy Yanish, who is the godfather of mouse transgenesis. Even he had to scratch his head when we were getting issues with Paramescus. But this is the more important thing. I've started putting this into my talks, public, particularly for the public. My lab's been funded by NIH since 1994. NIH grants have supported all of the major discoveries about tick-borne disease. Any attack on NIH is going to turn out badly for all of us. And so the public in particular needs to support the continuation of NIH and supporting this kind of very basic research that may have future public health implications. And with that, I have left a few minutes for questions.
Thank you so much, Dr. Telford. Yeah, anybody has questions, feel free to reach out, drop them in the chat, whatever works for you.
Hey, Sam, really nice talk. I really enjoyed hearing about that work. I've just been following the Oxitec work in Florida and the massive regulatory challenges they've had.
Yeah.
I mean, you touched on that briefly, but it seems maybe even more daunting here because at least
Yeah, well, we got time on our side, right? It's like when it was first introduced at the end, maybe mid 2000s, maybe 2010, it was an outrageous idea. And in fact, my former mentor, Andy Spillman, and Steve's as well, would have just been, he was just been horrified at the suggestion of doing genetically modified mice. He was known as an obstructionist for the genetically modified mosquito movement, saying, who is going to harbor a pest? We grew up generations saying mosquitoes are a bad thing, we must kill them. And now we're telling people we must not kill them, we must
We're
let
going to
them
release huge
proliferate.
numbers.
And he never saw that as being possible. And now we see the possibilities with genetically modified mosquitoes. Genetic modifications being used in all sorts of other applications, agriculturally, for example, the expression of vitamin D by rice. In Southeast Asia, a fantastic thing. And so I think 10 years from now, nobody's going to bat an eye about genetically modified animals. And if we phrase it correctly in public education, it's really no different than what we've done with dogs and cats. I mean, all of us have dogs and cats. We've artificially selected for traits that we like. There's no reason why we can't artificially select for traits that we like in nature as well. And so I think that's, that's a big sort of field where I'd like to see students and people put some time on, is educating the public about that new technologies aren't necessarily new. They're just variations on old what we've been doing all along. We're just finding more efficient ways of doing it. Instead of waiting generations for selection, we make selection happen overnight. Uh, but you're right. And a lot of people are like, well, the Lyme disease vaccine's coming. It'll be approved next year. Why do we need this for Lyme disease? And I fully admit this approach is like using a 10 gauge shotgun on a housefly. But I see past the actual public health implementation. At most, it'll be a tool. It'll be a platform in the event, although God knows when, that there are antigens and if there's antibody-mediated protection or reservoir competence reduction for things like Babesia or Anaplasma or Borrelia miyamotoi. Those cassettes could be put into this. Powassan virus would be a very low-hanging fruit. We know exactly how to make a vaccine. It would be very simple to do, except that white-footed mice don't seem to be the reservoir for Powassan virus. So, but nonetheless, the platform, I think, is valuable to develop. The concept of heritable immunization is important to develop. The actual implementation of this as a proof of concept experiment, I think we'll be able to at least get to a small field trial. Whether any community will adopt this after community informed consent, don't know. But it doesn't mean we shouldn't be doing this.
Dr. Rich, looks like you have your hand raised. You're still muted.
Sam, I just wanted to correct you on a factual error. I'm not 60 years old yet.
Oh, well, then I guess
I still have a few months.
Cause for commiseration soon, huh?
But I had a question about, have you started formally sort of looking at the ecology of what it would take to in terms of release? And are you basing it on some of the old literature suggests 95% attrition rate over winter for Peromyscus. Do you have better estimates on that, or have you started digging into that at all?
Yeah, I alluded to that, and obviously it depends very much on demography. Our strategy right now is going to be, if we get to the field trial, the isolated field trial, it would have to have release or inoculation. I'm still arguing that there's no way in hell that we can breed mice in the laboratory and release them into nature and have those mice have any kind of fighting chance. Although we do have an idea that, interestingly, Kevin put in a patent application for, where we'll use nest boxes that have RFID readers, and the nest boxes will have additional resources, like more food, and only the mice that were released will have RFID tags that will allow them to enter the boxes and access this resource, and thereby promote, at least if energy or if resources are limiting to Peromyscus populations, and that's a huge debate on its own. We'll have some way of promoting mice, at least the founder mice, but to hit them at the time when the population is at its lowest. Every year in every site that I've studied, barring one, spring is a cruel time. Winter and spring are a cruel time for mice. The population goes to 2 to 5 animals per hectare, and then over the course of the summer can build to 20 per hectare. And those 20, actually, it's my guess, we haven't done the genetics. We'd love to find a mammalian population geneticist to do this work if we can find the money, to show that, you know, those few founder mothers that survive in the spring give rise to the bulk of the 20 per hectare in September. I think that's a key data point that we need to demonstrate that this approach would work. But that's my approach based upon my knowledge of the demography of these mice. The one site where I don't see this is where there are no predators, there are no weasels, there are no snakes on all mice. The mice are very long lived on this little island in the North Fork of Long Island. Otherwise, you're right. You know, there's no way it'll work if we just take the crude approach and just dump mice willy-nilly into a site. And it won't be accepted by the community. We can't release thousands of mice. We're just going to make a lot of fat seagulls and hawks and things.
Just spitballing, but one thing that might be interesting to look at would be D-loop mitochondrial DNA as a marker.
Yeah, no, you did some of that very early on. I remember actually writing a grant with you on that.
And we could set the clock by some age structure looking at dentition.
Yeah.
Are you still trapping a lot annually?
Every summer.
Yeah. Just talk about that.
Yeah, I keep myself younger that way physiologically. My dad, he died 4 or 5 years ago, but his health declined very rapidly after he stopped doing field work. Well, at least I wasn't too controversial, or maybe I was so controversial no one has any comments.
Just a couple more minutes if anyone has any last questions. Well, if not, then thank you so much, Dr. Telford. That was a great talk. And I hope to see everybody in 2 weeks. On May 7th, we have Dr. Patrick Pearson, who will talk about Project Whitetail. So we'll see everybody then.